All right, hello. We're gonna run through nucleic acids and proteins today. So nucleic acids, you should know, hopefully, please know that there are two. There's DNA and RNA. DNA stands for deoxyribose nucleic acid. RNA stands for ribonucleic acid, okay? Regardless of whether it's DNA or RNA, there are three main parts. We have the phosphate group, which is in purple. We have the sugar, which is either ribose or deoxyribose, which is the blue. And then we have the base, which is pink. And there are technically 5 different bases that it can be. What else? Oh, also the phosphate and the sugar are linked by covalent bonds, as well as the sugar and the base are also linked by covalent bonds, which remember are bonds between nonmetals, okay? And then this image over here, if my pencil would work, this one right here, this basically just shows a single strand of, again, it could be a DNA or an RNA molecule, but just 1/2 or one single strand. So this would be a single strand. Okay. So we can see that the phosphate groups are usually represented by circles. The sugars are usually represented by these pentose, or these, basically, sorry, my phone was my watch was recording all of that. That was weird. So yes, and then we have the bases are represented by, excuse me, these rectangles. Okay. Again, this should not be new information. This should hopefully be a review for everybody, but just so we're all on the same page, the difference between DNA and RNA, the main difference is the 2 different sugars. Literally the only difference between deoxyribose and ribose is on what carbon is this? Let's count it out. I think it's 1. So this is carbon 1, this is carbon 2, this is carbon 1, this is also carbon 2. So on carbon 2, with DNA, there is no hydroxyl group, but with the ribose sugar, there is a hydroxyl group. Okay, literally the only difference between the 2 sugars. The second thing that's different is DNA is a double-stranded molecule. It's got a leading and a what we call we just call, for right now, we just say it's double-stranded. We'll talk about that in a second. RNA, however, is always single-stranded, as you can see in these pictures that I drew. And then, again, should be review. The bases, so remember the bases are the pink guys up here. The bases for DNA are adenine, cytosine, guanine, and thymine. And we can call them just by their first letter. And we know that adenine A binds to T and C binds to G with DNA. And then for RNA, we have, the only difference is instead of thymine, we have uracil. So in this case, A binds with U and C binds with G. Okay? This hopefully should be review. I believe Ms. Q talked about this last thing with you guys, but this might be the kind of newest information. So DNA, we know it's a double helix, and it's made of 2 anti-parallel strands. So the strands, strand 1 is basically going the opposite direction of strand 2 in DNA. And the bases, as you can see in this picture right here, the bases are linked by hydrogen bonds. Okay, and so when we say anti-parallel strands, sometimes you will also hear 5 to 3 direction. So, you know, one strand, we're going from the 5 to 3 direction, whereas the other strand goes from the 3 to 5 direction. And where we get those numbers from are basically from the carbons. So remember, if we count carbons, if you look at this picture right here, this is what it would look like if you were looking at it kind of like on the molecular level and actually looking at all of the elements. So if we were to count carbons, I don't want that color. What color do I want? Sure, let's go with pink. So again, I kind of counted it out for you, but look, we have carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. So carbon 5 is bonded to the phosphate group. And then carbon 3 down here is also bonded to the phosphate group, but it starts with carbon 5. We start with carbon 5 at the top, like right here, and this is carbon 3 right here. This would be carbon 5, this would be carbon 3. Carbon 5, carbon 3. So that's why we say it's the 5 to 3 direction. If we were to look on the other side, we would basically imagine that you're like flipping it over. It's literally like the same thing, but upside down. So this is carbon 3, this is carbon 5. This is carbon 3, this is carbon 5. This is carbon 3, this is carbon 5. This is carbon 3, and so on. So it starts with carbon 3 right here, and then it goes from carbon 3 to carbon 5. And like I said, over here, we start with carbon 5, and we go to carbon 3, okay? So I just want to make sure you understand why we say 5 to 3 direction and 3 to 5 direction, but basically they're anti-parallel to each other, so they're basically flipped. One is upside down. And you also need to know that the bases are linked by hydrogen bonds. The last thing that I'll mention, you guys do have to know about Watson and Crick. You should already know about Watson and Crick. They basically made a model of DNA. But we also know that without the discovery of Rosalind Franklin's image 51, so she, and this is actually a picture that was taken by, I believe, one of her graduate students as well, but she was doing her own research on DNA, and so her graduate student took a picture of this. Watson and Crick literally saw it on her desk and came to their aha moment, because as you can kind of see, it's kind of hard to tell, but you can kind of see these, like, basically it would be like if you're looking at the helix from the top. So I wish I could show you. I'll show you in class because I have a DNA model, but this would be like if you looked at the DNA model from above. And so they basically saw this picture and they were like, aha, it's a double helix. And so they didn't credit her or her graduate student, and you know, there's that. So you have to know about them, but you know, you already know about them. So that's all for nucleic acids. Hopefully it's mostly review. Okay, on to proteins. All right, proteins. We're gonna run through this. Some of this may be new information. We're gonna go a little bit more in depth with proteins, but a lot of this, again, should be review. So the monomers of proteins we know are amino acids. They're linked together, and this is actually not written down, so I do want you to add this. So amino acids are linked together using peptide bonds. Peptide bonds. So the bonds that form between amino acids, we call them peptide bonds. And amino acids are linked together to form polypeptides. Poly meaning many, so many peptides. Again, we should remember that any kind of reaction that links together molecules to make a macromolecule, so that's a condensation reaction. Hopefully you said it in your head, condensation reaction. Which is also anabolism. Okay. Polypeptides then get folded to make proteins. And then some proteins can be single amino acids, or they can also be multiple amino acids. Sometimes proteins have more than just amino acids, but for the most part, just understand that polypeptides are, sorry, it's been a long day and my brain is tired. I'm going to keep going, but basically you need to understand that sometimes proteins can be single, like it can literally be a single amino acid, but then sometimes you have lots of amino acids. Cool, great, moving on. You need to know how to draw a peptide bond and basically how it forms. So if we look at this, and I know it looks a little crazy, so we're going to look, hold on. Oh my gosh, like I said, I'm so tired. Okay, this part, okay, this is basically, we have amino acid 1, okay, right here, this is amino acid 1, amino acid number 1, and this is amino acid number 2, and we are combining them, okay? So first thing that we need to know is we need to know how to draw an amino acid. And when I say draw an amino acid, we're drawing a very simplified version of an amino acid. So you need to know that an amino acid has essentially 3 important parts. We have this region right here, which is the amino group. Okay, so the blue represents an amino group. It's nitrogen that's bonded to 2 hydrogen. We have on the other side of the molecule, we have in the purple, we have a carboxyl group, which we've already looked at because remember lipids also have a carboxyl group. And then we have an R group. And we just use R as a catch-all for whatever amino acid. So basically every R group is different depending on what the amino acid is. So instead of, you know, doing a big old thing, we're just gonna use R to represent like the unique part of the amino acid, okay? So to form a peptide bond, remember this type of reaction, I already said it above, but it's a condensation reaction. Condensation reaction. And so remember, a condensation reaction, we're going to form water, okay? So where the water comes from, we have an OH over, my pen keeps stop working, anyway. On the carboxyl group of the first amino acid, we have the OH, okay? And then on the amino group of the other amino acid, we get this H. So we have an OH over here and another H over here, and those will form to get the water. So the actual bond is right here between the C and the N, okay? So we will do some practice with that, but you do need to know how to draw, like I said, a peptide bond, okay? You will basically you will need to be able to draw this, okay? All right, so like I said, there are amino acids. Amino acids are the building blocks of proteins. They're the monomers of proteins. And you do need to know that there are 20 different amino acids. I took this straight from the IB Bio textbook. You don't necessarily need to know this, but you do need to know that amino acids are very diverse. So there are 20 of them. Some of them are hydrophobic. Remember, hydrophobic means that they are insoluble in water. Some of them are hydrophilic, so they can dissolve in water. Within the hydrophobic ones, some of them contain rings, some of them do not. Within the hydrophilic groups, some of them are charged, meaning that they have like a positive or a negative side. And some of them are polar but never charged. So lots and lots of different types of amino acids. And they all kind of have like their unique role, okay? And amino acids can be linked in any sequence, which means that there are many, many possible sequences. So there's a lot of polypeptide diversity. So this is, again, straight from the textbook, but this is just to kind of show you all of the possibilities. Like, even if you only have a protein that is made of, like, let's look at this one, for example. Let's say you have a protein that's literally one amino acid, okay? Even if it's only one amino acid, you have 20 different possibilities, okay? Because we can do all 20 different amino acids, okay? If we had 2 amino acids linked together to make a protein, there are literally 400 different possibilities. So, you know, for example, we can have, you know, I'm trying to think, like valine and lysine. God, I can't, lysine, okay? Or we can have lysine and then valine, you know? So basically the way that we figure this out is we take the number, there's 20 amino acids, and we multiply it, or we put it to the power of like whatever number we have. So if we had 3 amino acids, we would do 20 to the 3rd power, or 20 cubed. So we do 20 times 20 times 20. So even if we only have 3 amino acids, that is 8,000 different possibilities, okay? And all of those possibilities would be unique, okay? So if we continue to fill in this chart, you know, if we, let's say we had 4 amino acids, that'd be 20 to the 4th. And you can do this with me. I'm literally doing this on my calculator right now. Oh, I should have had this up, but I didn't. Calculator, and I'm just doing it on my phone. So 20 times 20 times 20 times 20. All right, 160,000. Again, only 4 amino acids, and look how many different possibilities we have. This one is easy. If it's 20 to the 6th, that means there's 6 different amino acids. And I already did the math for this. I believe it is if there were 10 amino acids. So if there were 10 amino acids, that'd be 20 to the power of 10, which basically means, again, we're only talking about 10 amino acids. There are 10.24 trillion different unique possibilities in terms of like the order of the amino acids and what amino acids are included. So even though there's only 20 different amino acids, there are many, many, many different possible sequences. So that's what makes proteins so diverse, okay? Then we need to talk about the central dogma. This is like the be-all, end-all of biology, essentially. So we're all familiar with DNA. We just talked about DNA. So DNA is transcribed. I'm going to write this in a different color. Let's do, I don't know. Let's go crazy. Nah, it's too dark or too light. That's too close. God, my brain. Listen, it's been a day. Okay. So DNA, we say that it is transcribed. We'll talk about this. Transcribed. So DNA is transcribed into RNA, which is then translated into proteins. So again, this is why we're talking about the biomolecules or macromolecules. It's all connected, okay? DNA codes for RNA, which codes for proteins. So they are literally essential to life. Without DNA, you wouldn't have proteins. Without proteins, and we're going to talk about this in a second, proteins are used everywhere in your body. And the function of proteins is incredibly diverse. Again, because there are so many different possibilities of like how many different amino acids and what the structure can look like, okay? So going back to this, every individual has a unique proteome. I know I'm saying that wrong, but whatever, it's late, whatever. So the proteome, similar to like a genome, okay? So proteome is all of the proteins produced by a cell, tissue, or organism, okay? So it's not exactly a genome. So a genome is like all the DNA, it's all the genes. But for a genome, not all of the genome gets translated into proteins. So you may have a lot of DNA that doesn't code for a protein. So that's why a proteome is different than a genome, because there's a lot of, like I said, there's some DNA and RNA that is non-coding. Okay, so this should be a new term for us. But proteome just means all of the proteins that get produced by a cell, tissue, or organism. Usually we're talking about an organism. All right, almost done. Protein conformation, so basically like the 3D structure of proteins. There are two main conformations. We have fibrous proteins and we have globular proteins. So fibrous proteins are elongated. You can see that they are long and like very, they're very long. That's what I'm trying to say. They're elongated. Globular proteins are more compact. They kind of look round. And so again, fibrous proteins, like it kind of like the structure relates to their function, right? So fibrous proteins are usually used in structure and to make things, they're usually provided for structure. I don't know. Globular proteins are more like enzymes, and they are not for structure, and they're for other things. And so the last thing that you need to know is that, and we'll talk about this more when we talk about enzymes, but you need to be careful with proteins with extreme temperatures and pHs. So if it gets too hot, too cold, or if it gets too alkaline or too acidic, then this can irreversibly change the structure of the proteins, and basically the proteins can fall apart essentially, or they can sometimes even be rearranged and like reconfigured, which will then affect like the function. So we call this denaturation. Denaturation. God, I'm so sorry. I'm never doing it after this long day again. God bless. Okay, moving on. Last thing, protein functions. Again, proteins are in everything. So you can kind of look this over on your own. It's in your notes. I'm just kind of going to go over like the important ones that I know that we'll definitely talk about. So we'll definitely talk about like this is essential because we're going to talk about this next. So proteins are catalysts. Enzymes are catalysts. Enzymes are a specific type of protein. They catalyze or speed up a reaction. We got muscle contraction, cytoskeletons, tensile strengthening. We'll talk about this with collagen. So, and I talked about it with like the fibrous proteins. So a lot of fibrous proteins are used for tensile strengthening. So it gives strength to skin, tendons, ligaments, etc. Blood clotting. So without clotting factors, which are proteins, then you wouldn't be able to, like, you would just keep bleeding forever. And that's why people with certain genetic diseases, they have issues with their clotting factors, and that can be real bad. Transport of nutrients and gases, we'll talk about probably next year for you guys when we talk about human anatomy and physiology. We'll talk about hemoglobin. Hemoglobin is a protein that transports oxygen in the blood. Membrane transport's another important one. There are proteins like channel proteins that let certain molecules in and out of the cell. There are proteins involved with like active transport. Hormones, so not all hormones are proteins. This is really important. Some hormones are lipids, but in particular, insulin is a protein that you do need to know that is a hormone. And it is, like I said, it's a protein. So insulin, we'll talk about what insulin does, but hormones are essentially chemical messengers. Packaging of DNA, histones, we'll talk about this, but histones basically help package DNA in eukaryotes, and they help chromosomes condense during mitosis. And then proteins are also necessary for immunity because of antibodies. Like antibodies are proteins. Okay, so very specific. You do need to know all these 4 proteins and specifically their function. So Rubisco is known for, it's specifically in plants, and where is it? It helps catalyze the reaction that fixes carbon dioxide from the atmosphere. So without Rubisco, plants wouldn't be able to take carbon dioxide and basically make it into glucose, okay? Insulin is, like I said, a hormone. It is a signal to many cells in the body to absorb glucose and basically reduce the glucose concentration of the blood. Immunoglobulin is a globular protein. We talked about those above. They're globular. Immunoglobulin is an antibody, okay? And they basically bind to antigens on bacteria or other pathogens and basically like help destroy them, okay? So we need antibodies. Like when we have COVID, we develop COVID antibodies that can basically track and basically bind to the antigens of COVID, the COVID virus, and cause the response that basically gets them destroyed. Rhodopsin is a pigment that absorbs light, and it basically detects light intensity. It's in the where is it? It is. Yeah, it leads to the rod cells sending a nerve impulse to the brain. Hold on. But yeah, it's in your, it's a membrane, there it is. It's a membrane protein of rod cells of the retina. So it's in your eyes. It's a pigment that basically detects light intensities. Collagen is specifically in animals and humans. It's a mesh of fibers in the skin and blood vessel walls that resist tearing. So it basically keeps your, it gives ligaments and blood vessels their strength. And then we have spider silk, which is basically the silk that spiders produce to create their webs. And it is very, very extremely strong. Even sometimes some types of silk are stronger than steel and tougher than Kevlar. So the silk is very stretchy, it's extendable, very resistant to breaking. Okay, so you need to know all those 6 different types of proteins. All right. Oh my God.